Right, so we will talk about the histology, what the slides that you are going to see, slides that you're supposed to be able to identify with regard to cartilage, bone, and also muscle, right? So a small timetabling error has put the practical before the lecture, but that is all right. We'll just learn the histological features of the muscle in this practical, and then you will have a lecture next week where you will be learning in more detail the features of the muscles, right? So at the end of the practical, you can ask questions with regard to the lectures that were done, also the practical today. Okay, so this you know already. We classify cartilages according to the amount and type of fibers that are found in the matrix of each of the cartilages. And accordingly, we divide them into three types: hyaline, elastic, and fibrous. So hyaline cartilage has a large amount of cells and a small amount of collagen fibers, while the elastic cartilages has a moderate amount of cells and a large amount of elastic fibers. Then fibrous cartilage has few cells and a large amount of collagen fibers, right? So according to the things that are found in the embedded in the matrix, the amount and type of the fibers, you get different features to the cartilages. So these cartilages are designed to perform different functions. There are slight changes in their functionality according to the amount and type of the fiber. Okay. Right. So if you take cartilages in general, this is of course a hyaline cartilage. Now here you can see the perichondrium, right? So as I told you in the lecture, we have the perichondrium. Now these pink staining areas, those are the connective tissues, and you can see small cells, right? Tiny cells here. Those cells are the fibroblasts, right? So those fibroblasts have the ability to convert into chondroblasts. These are the chondroblasts that you see in the periphery. Okay, so the fibroblasts in the perichondrium can turn into chondroblasts, right? So the chondroblasts, you can see they are single cells, okay? Single cells. So you can see them as cells. You can see the lacunae actually. What looks like the cell margin is actually the lacunae, which is basically a small tiny space between the cell and the matrix. Now this is the matrix. It looks just white, right? We call it amorphous because it doesn't have any character, right? So we say it's characterless. And you have collagen fibers in here and the matrix proteins, but all of those have the same refractory power. Because of that, when you shine a light through it, when you do a staining, you don't see anything. You just see it as a sort of a clear area, right? So some areas may be more darkly stained if there is more protein, more matrix proteins. Other areas lightly stained, right? So just a plain sort of color. Okay, so depending on the amount of stain, it can either be whitish or light purple, light pink, that kind of color. Okay, and then as you come towards the center of the cartilage, move towards the center, you can see groups of chondrocytes. Yeah, so inside you can see the nucleus of the chondrocyte. Okay, those are the small nuclei, and the cytoplasm is seen in white color, right? So in the middle of the white color, you see a dot of dark purple, that is the nucleus, right? So these chondroblasts divide and give rise to more and more chondroblasts, and you see groups of cells. Generally, you can see 3 sometimes. 4 is difficult because when you cut it, you won't get all 4 cells. Here, I think there is one here with 4, right? So like that, you get groups of cells because of mitotic division, right? So a single cell dividing into 2 will give rise to 3 cells. Then those 2 again dividing will give rise to 4 cells, right? And again, sometimes one of those cells may again divide, giving rise to 6 Like that, you get groups of cells as you go towards the deeper areas of the cartilage. Okay, so you have to observe the perichondrium, which contains blood vessels. Here, of course, you can't see blood vessels clearly, but you can see the collagen fibers and the fibroblasts. Then the amorphous matrix, single chondroblasts in the periphery, and chondrocytes in groups in the deeper areas, right? So if you are given this slide and asked what it is, if you say identify the tissue, you will say cartilage, or you can say hyaline cartilage also, right? So you say hyaline cartilage. Then the second question may be give reasons for your identification. So you will say there is a amorphous matrix, there are chondroblasts, single chondroblasts in the periphery, and chondrocytes in groups in the deeper layers. Also, the perichondrium can be seen. Okay, so if you write like that, you get full marks. Okay, right? So this is a slide that is there in the lab where if you get a chance, you will be able to see them, right? And it is an H&E section 10 to the power 10. Okay, so collagen fibers in hyaline cartilage is not visualized in H&E section, right? So so that is how it looks like. So this is a low power view, 10 to the power 10. Okay, and then coming on to elastic cartilage, right? So this is the epiglottis, cartilage taken from the epiglottis, a section of the epiglottis done in a special stain to get the elastic fibers stained. Okay, so this stain is called Verhoef's. You don't have to know the names of the stains, right? These are not necessary, but remember to be able to identify it either as a special stain or a H&E stain. Okay, so naming the stain is not that important. Okay, right? So this section has been stained with a special stain so that you can see the elastic fibers which have been stained in black. Okay, so the black stain is there. All these areas in black are the elastic fibers, right? And you can see quite a large amount of cells, moderate amount of cells, right? And around that, the matrix, and in the matrix, you see lots of elastic fibers, right? So the extracellular matrix or the cartilage matrix or the cartilage extracellular cartilage matrix will have these elastic fibers, right? So these elastic fibers give you the elasticity that it requires, right? So if you want to, if you look at your ear, you can bend it different ways, right? So that property of elasticity is given by the large amount of elastic fibers that you find in the matrix, right? And then of course you get chondrocytes. Here you see only chondrocytes because this is the middle of the cartilage that you see. So chondrocytes appear lightly stained with a central nucleus. So that's, sorry, so that would be a nucleus, right? Those tiny dots in the middle are the nuclei. Okay. Right, so then we don't have a very good slide of fibrocartilage. Fibrocartilages are a little bit difficult to find, right? You can get them in places like inside joints, joint capsules, in tendons, some ligaments, you get small discs of fibrocartilage, right? So in the morning, we talked about joints, and in the epiphysis, you will find a disc of fibrocartilage, right? So it's a little bit difficult to figure out whether it's a cartilage because cells are minimum and there's lots of condensely packed collagen fibers, right? Because of that, it is a difficult slide to identify properly, right? However, there are different, I have shown you pictures which are much better than the slide that we have. This is the picture of the slide we have, but you have seen pictures of slides which have been taken from the net, right? So in the lecture, you saw better pictures of fibrocartilages, right? So scattered chondrocytes, or actually sometimes the chondrocytes may be arranged in the lines of stress. Even the collagen fibers are arranged in the line of stress, though it is not visible in this slide. Okay. right? So then talking about the articular cartilages. Now, articular cartilage has a very special sort of appearance, and it has several different layers. It is a specialized form of hyaline cartilage, as I said, and it does not have a perichondrium surrounding it. So its ability to heal itself is minimal. Okay. So what are the 4 different layers? You have a tangential layer, okay, transitional zone or layer, radial zone, and the calcified cartilage layer. Okay, so these 4 layers are seen in articular cartilage, and the articular cartilage, as we learned earlier, is continuous with the underlying bone. Okay, so the underlying bone can be observed below the calcified cartilage layer. Right, so these 4 different layers are actually seen according to the chondrocytes, which are arranged in different patterns in the articular cartilage. Right, so from the superficial to the deep layers, the chondrocytes are arranged in different manner so that you get these different layers to the articular cartilage. It is one of the thickest cartilages that you see, and it is very important in the synovial joints. Okay, so I think I showed you this before also, but here in this is also a special stain, and this is extremely low power view. We use 10 to the power 4, right? So because it is a thick tissue, thick cartilage block, you can't see it in a high power view, so you have to look at it in a low power view, right? So the 4 layers can be seen. So initial small flattened chondrocytes in this area, okay? And these lie parallel to the surface of the cartilage, right? Small flattened ones parallel to the surface. Then transitional zone, chondrocytes become slightly larger. You can see them. These small white colored dots are the chondrocytes, right? So transitional layer is there. You can see them either single or in isogenous groups, right? 1 or 2 cells, right? And then you get the radial zone where the chondrocytes are large and they lie in sort of like lines, right? So several chondrocytes lining up like that, okay, right? And they are perpendicular to the surface, right? And then at the bottom you can see calcified area. Now the blue stain is for the calcium, and the pink stain is for the protein in the cartilage, right? So glycosaminoglycans, chondroitin sulfate and keratan sulfate are stained in pink, and the calcium is stained in blue, right? So here at the end, the calcified zone, you can see that some parts of it is blue, right? So this area is a calcified cartilage, and this, of course, is the bone, and this is the bone marrow, right? So the bone is stained in bluish blue, right? Bluish greenish color. Okay, so this calcified layer rests on the underlying cortex of the bone. All right, so this is actually articular cartilage, again has to withstand a lot of stress, and that is why you have these cartilage cells in different ways, right? So they are, because of the stressors, the cartilage, the chondrocytes lie in different ways, right? So either they are parallel to the surface or they lie perpendicular to the surface in radial columns, right? So this arrangement helps the articular cartilage to withstand a lot of stress and be free of friction during all the movements of the limbs. Okay. Right. So this is a slide that we have of the epiphyseal growth plate. We have quite a few nice slides of that, right? So you can see. And you can see very clearly the 6 different regions. Okay, so firstly you can see the zone of reserve cartilage or the resting cartilages here. Okay, and then you can see the cartilage cells dividing, proliferation stage. Okay, and then you can see the zone of maturation where the cartilage cells are becoming larger and larger, right? Maturing, and then you can see them increasing in size. This one here, okay, increasing in size, right? And calcification starts to occur. Again, here the darkly purple areas are the areas where there is calcium. Okay, so the light purple area, almost whitish area, is where you get the matrix proteins of the cartilages. Okay, and then here you can see cartilage degeneration because the matrix is calcified. The cartilage cells are dying here, and then finally you can see the bony areas here. Sixth one, right? So that is the bone area here. Dying cartilage, increasing in size, maturation, proliferation, sorry, resting. Okay, proliferation is this. Okay, really messed up, right? Okay. Right. Okay, you didn't get confused, I hope, right? Are you okay with that? I put the arrows in the wrong places and I don't know how to get this off. Wait a bit. Okay. The eraser is not working apparently. Okay. Okay, are you okay with that?
Yes, madam.
Okay, I hope you didn't get confused, right? So the numbers are correct. I just put the arrows wrong, right? Okay. Right, so this is just the description, right? So I've gone through this many times just to summarize. So you have the reserve cartilage, which is hyaline cartilage with the chondrocytes arranged in clusters, just like you will see in any hyaline cartilage anywhere. Then the zone of proliferation where mitotic division happens and the chondrocytes are seen in columns or groups, right? And then maturation, cell division stops there and the chondrocytes starts to enlarge and mature. Okay, and then the zone of hypertrophy and calcification where large chondrocytes are there and their cytoplasm gets filled with vacuoles of water, right? And surrounding that, the cartilage starts to calcify. Okay, and then zone of cartilage degeneration where the chondrocytes degenerate and die basically, and osteogenic cells and capillaries from the bone marrow invade into these spaces, and those osteogenic cells start to differentiate into osteoblasts and divide and lay down bone matrix, right? So the rest of it becomes bone. Okay, right. Okay, then compact bone. We talked about different types of bone. So this is a type, the thickest and the strongest type of bone, a lamellar bone, Havasian system. This is the compact bone, right? So the compact bone, if you take a slice and stain it with HNE, you won't be able to see much of it, anything, right? So what you do is you take a thin sliver of bone and grind it between two stones, okay? Special stones until it becomes really thin, and then you can just observe it. It is not necessary to stain it. So this is such a thing. We have lots of slides of this. So this is a compact bone ground section, right? So here you can see the Havasian canal in the center here, right? And these black dots are actually the lacunae where you will get, where you would have had your osteocytes, right? So when the bone is ground, the osteocytes die actually, and they are no longer there, but these are the lacunar spaces, right? So that is the Havasian canal, and that is the Walkman's canal, and you can see the concentric layers of the osteocytes. Okay, right? So Havasian canal in the center, concentric layer of mineralized matrix of bone, as well as the lacunar spaces. Then the Walkman's canals, right? So this is prepared by grinding a small piece of bone as thin as possible. No staining is required, right? So this is again low power view, 10 to the power 10, no staining. Okay. right? So here you can actually, this slide is specially made to look at osteoclasts, right? So you can see osteoclasts, several osteoclasts actually, and you can see the other ones also, right? But here, this is osteoclast, right? So you can see that it is inside this cave, okay? These osteoclasts. Okay, so you can see several nuclei inside. Okay, so osteoclasts, they have multiple nuclei, as I told you, and they are inside this small depression, which we call the house-shaped lacunae. Okay, so this is important in bone remodeling and involved in dissolving calcium from the bone when it is necessary, right? So we discussed this in the previous lecture, and they are controlled by hormonal activities, right? So this house-shaped lacunae, and the this is also osteoclast. This one osteoclast, right? So actually these cells tend to be in groups, and if you look at the cytoplasm, you have several nuclei inside it. They have all these properties of macrophages, right? So macrophages are derived from the monocyte cell line, and they have the ability to engulf particles, right, and destroy cells. So similarly, osteoclasts have the same they come from the same lineage, and they have the ability to destroy bone completely, the organic as well as the calcified parts of it, right? Okay, so then looking at cancellous bone, again, if you do a H&E section, this is what it looks like. Most of it is pink, very difficult to see the nuclei. You can see the nuclei, I mean, they're not very clear, but you can see them. And you can see the endosteal osteoblast layers, right? So endosteum means the lining of the inside of the lamellar bone. The lamellar bone, in the sense spongy bone, right? So cancellous bone, a spongy bone, which has inside it, you have the pink staining trabeculae of the bone, right? So this pink area are the bone areas, right? Rest of it is the marrow. So this is the endosteal layer, and this is inside is the marrow. Okay, so osteoblasts are there in the endosteum at the edges, periphery. Okay, so the bone marrow is seen in intervening spaces. Now, as you know, bone is a highly calcified matrix, so you can't put it into that microtome that we looked at. Okay, you can't put it in that, you can't cut it, right? So if you want to cut a section of bone with the microtome, then you have to get rid of this calcium. So it is decalcified with a chemical called EDTA. Okay, and then only you section it and stain it with H&E. All right. Okay, so here you can see actually this is a fibula actually, fibula which has been decalcified and tied in a knot, right? So this bone, all the mineral inside this bone has been dissolved by using EDTA, right? So if you place a bone inside a solution of EDTA for a long period of time, all the calcium gets dissolved, right? So then that just shows you that this hardness in the bone is given by the calcium salts in the matrix, right? So the proteinaceous substances and the other parts, cells and all that, actually does not have that. So it can even be tied in a knot. A bone can even be tied in a knot when you remove the calcium from it, right? So this specimen is there in the lab. You can see it when you come. Right, so then I will just talk about the muscles as well, even though we haven't done the lecture, right? So you have three types of muscles, skeletal, smooth, and cardiac. I'm sure you know this, right? So skeletal muscles are the, skeletal means they are the muscles which are attached to the bones, right? So all the muscles in the limbs are skeletal muscles, right? And then smooth muscles are the muscles that you see in the organs, right? So like the walls of the gut, the body of the uterus, then the walls of the blood vessels, right? All those things have smooth muscle lining them, right? Then of course, cardiac muscle is a highly specialized muscle, which is seen only in the heart. Okay, so if you take a section of the cardiac muscle, this is a H&E section, high power view, 10 to the power 40. You can see the features of the skeletal muscle, right? So the skeletal muscle is fiber is cylindrical, right? It might appear as if this is branched, but it's not, right? So you get a cylindrical muscle fiber like this, not branched. You have cross striations. Okay? Cross striations like this, and many branches, close, right? Close cross striations, and multiple nuclei, multiple peripheral nuclei, right? Peripheral, multiple nuclei, and those are the nuclei, right? So you generally call a muscle cell a muscle fiber, okay? Right, so these are not actually, these are the fascicles that have been cut from different areas. That's why it looks like this, but it's not branched, right? And you can vaguely make out the cross striations there. So these are the cross striations and cylindrical shaped with peripheral nuclei, right? So cross striations, peripheral nuclei, multiple nuclei. So if you're given a slide of this, then you're asked to identify the tissue, you say skeletal muscles. If you're asked why, you say it is a cylindrical shaped cell, unbranched, and there are peripheral nuclei, and the cell is striated. There are cross striations, right? So peripheral multiple nuclei, cross striations, and unbranched cell tells you that it is a skeletal muscle, okay? Right, so skeletal muscle, if you do, sorry. This is a, it's written here, right? Okay, so again, if you want to look at this cross striations properly, I mean, if you put 60, it looks even, you can see almost all of the cross striations, right? So here you use a special stain which allows very clear visualization of the cross striations, right? So here we have used a special stain called PTAH and high power view, 10 to the power 40, to look at the cross striations, right? So you can see the darkly staining areas, the lightly staining areas, and all those things. Actually, when you learn about the actin myosin filaments and the cross striations, you can, if you look very carefully, you can see those, right? However, to see all of that, the sarcomeres and all that, of course, you need an electron micrograph, right? Okay. Okay. Right, so then smooth muscle is basically seen anywhere in the gut. If you take the wall of the gut, you can see them. So they are consisted of small elongated spindle-shaped cells, right, and tapered at the edge, right? So the cells would be something like this, like a fibroblast, right, flat nucleus. So you have these smooth muscle cells like that. Okay, so they have tapered ends. Sometimes they can be bifurcated like this, okay? And then it looks a bit like they're branched, but they're not branched, okay? So there is a single centrally located nucleus, and there is no striations, okay? So irregularly branching fascicles may be there, right? Arrangement varying from one organ to the other. Fascicles meaning like you have areas of smooth muscle like this, like that, okay? So you have cells like going like this. This word fascicle sometimes confuses students, right? There's no branching, it is the fascicle appearance, right? Sorry? Okay, right? So, so these smooth muscles can be seen, as I said, in the intestinal wall and in different blood vessels, right? Lymphatics, blood vessels, all those places you can see the smooth muscles, right? So you will learn about the muscles in the lecture next week. Okay. right? So the next one is the cardiac muscle. It is a specialized muscle and we call it the heart muscle. It is only present in the heart, right? So again, to look at the striations, we have used PTH here, special stain PTH, right? And then because of the PTH, we can see the cross striations very clearly, and cardiac muscles are of course branched, okay? And the most important thing are these intercalated discs, right? So PTH actually, you can see the intercalated discs very clearly. Okay? So these are the intercalated discs which are visible, and of course you can see the cross striations. Right? Cross striations and intercalated discs, and there are branchings. Okay, so intercalated discs are, as I said, junctional complexes that connect each of the cardiac muscle cells together, right? So these intercalated discs and the branching pattern of the cardiac muscle cells help the cardiac muscles to contract as a single unit, right? So the heart has to contract in a single unit. The ventricles or the atria, they have to contract at the same time, right? So there has to be a good communication between each of the different cells. And because of that, this communication between the cells are important. Okay, so you have a cell like this connected to another cell. Right, so intercalated disc could be there and another cell would be there. Like that, okay, and a central nucleus. Right, sometimes there may be one or two nuclei, right? But generally it's one. Okay, so if you're given this and you say, what is the tissue? You say cardiac muscle, and why you say it's striated, it has single nucleus, it is branched, and there is intercalated discs. So branching and intercalated discs are seen only in cardiac muscle, right? So if you actually asked for one feature or identifying feature of cardiac muscles, what you can say is that intercalated disc, right? So intercalated disc is the one that is specific to cardiac muscle. If you ask for a specification in skeletal muscle, it would be peripheral nuclei, right? Multiple peripheral nuclei, because striations are there in cardiac muscles as well. Okay, right. So branching and intercalated discs only in cardiac muscle. Okay, so this is the last slide. This is where you see the motor end plate or the neuromuscular junction, right? So this is again a special stain. It is a stain which uses heavy metals, right? And to stain the nerve and to stain the nerve endings. Okay, so this is basically in black. You see the nerve, okay, and it branches out and ends at each and every muscle fiber, okay? And then where it ends, you have the motor end plates, these tiny areas here, right? So I'm sure you have seen pictures of the motor end plate where it ends like this on the muscle cell, right? So you have all these vesicles at the bottom like that. So communication between the nerve and the muscle fiber, there is a small gap here, and that gap is the signal from the nerve to the muscle is traversed. The gap is traversed by chemicals, right? So this part is called a motor end plate, right? So it is seen, these are all skeletal muscle fibers, right? And the branched nerve stained in black is visible, and the motor end plates ending at all the different muscle fibers, right? So single muscle fascicle will be supplied by a single nerve fiber. It will branch out and supply each of the muscles, right? So that particular area will contract together. Again, the nervous control of these skeletal muscles, all the fibers together is necessary, because otherwise you will have something called fasciculations where different muscle fibers twitch or contract at different levels, but that will not be functional, right? So in order to give a proper contraction and move the limb, you need to have a coordinated contraction, right? So all the muscle fibers will be coordinated by these junctional complexes or motor end plates or neuromuscular junctions. Right? Okay, so those are the slides that we have, and that is the end of the practical for you. So you can ask questions if you like. You can type them in the chat box. Or just ask questions also if you want. Right, I'll give you a little bit of time. If there are no questions, we can stop.
Excuse me, madam.
Yeah.
Can you explain again, please, the physical appearance of smooth muscle?
Physical appearance of smooth muscle. Okay. You will, another lecture will be there, right? So it's just that fascicle means groups, right? Groups of smooth muscle. So you have, if you say like this areas like this. Okay, so areas, now this also is a fascicle. Okay, areas of smooth muscle like this. And there, inside those, you have the cells, smooth muscle cells. Okay, so these areas may be separated by connective tissue also, like groups and groups of muscles. Okay. So again, in the lecture also you will get this. So muscle fascicles are actually groups of cells, right? So you might actually say fascicle for skeletal muscle also, right? Not for cardiac usually. Smooth muscle also, small groups of muscles. So you see a layer of smooth muscles. Within that layer, you see the groups sort of divided into small areas like that. Okay, so inside only you get all the smooth muscle cells, right? So cells are arranged into fascicles. Okay.
Okay,
So
madam.
there is no, yeah. So you don't mistake it. Only the fascicles are branching, right? Not the cells. Cells are non-branching. They're single, elongated, spindle-shaped cells, right? Okay. And then somebody wanted the last slide. Yeah, so this slide shows you skeletal muscle fibers. All these brown ones that look like seaweed, those are skeletal muscle fibers, right? And this one here, the black one, is a nerve fiber coming down and branching out, right? So it branches out and reaches each and every one of these muscle fibers, okay? And then the ends of each of these nerve fiber goes and sort of grows into sort of like a small button-like area here, okay, at this end, and those are the ones that are seen there, okay? So actually seeing it will look like this, and there will be tiny dots like things, right? Yeah, you won't be able to see this membrane here. So the nerve fiber and these small dots, okay? So that is, you can call this a terminal bouton also, or the motor end plate, right? Motor end plate, or a neuromuscular junction. So a junction between a nerve and a muscle, right? So skeletal muscle and a nerve fiber. And in this area, you have these membranes here. So this gap here is called a synapse. Okay, so you will learn this. I think you must have learned it in when you did neuro. I mean, you must have learned it in A-levels definitely, but neuro, I think you had a lecture. So nervous system introduction. So this is a synapse, a gap between the nerve and the muscle, right? So synapse is bridged by the chemical, whatever chemical that may be there, right? Is that all right? Somebody wanted the last slide.
Yes, I got it, madam.
Okay, right. Okay.
Excuse me, madam.
Yeah.
In the morning section, we learned distal tibiofibular joint is a fibrous joint. What
Yes.
about the proximal tibiofibular joint?
Yeah, proximal one is a synovial joint, right? So there's not much movement there, but still it's a synovial joint. Okay?
Okay, madam. Thank you.
Right, we just see the... Okay, a lot of questions and then Right, just see. Last slide I explained. Taper, dense, and bifurcate. Yeah, so the cells are not bifurcated. Smooth muscle cells have taper dense. The cells have taper dense, single nuclei, right? They look like squamous cells, basically, but it is a smooth muscle cell. And the smooth muscle cells are arranged into fascicles. Fascicles may look irregular and branched, right? Cells are not branched. There is no branching in smooth muscles, okay? Okay, what basis do you classify primary and secondary cartilaginous joints? Yeah, so primary cartilaginous joints, secondary cartilaginous joints it's a histological classification, structural classification. In the primary cartilaginous joints, there's hyaline cartilage and bone, and there is no possible movement whatsoever. In secondary cartilaginous joints, there's hyaline cartilage and fibrocartilage, and maybe a tiny cavity, and there is some movement possible. Okay. Stain used in fibrous cartilage. Yeah, so fibrous cartilage is actually you can use a special stain for collagen, but fibrous cartilage, that particular one is a special stain, but it is not very good, right? That slide is not very good. But for collagen, if you want, you can use a special stain. Otherwise, it is just stained in pink if you do HNE, right? So if you do Verhoef's, actually, even for fibrous cartilage you can do, but you will see there are some stains which stain collagen in brown, right? So you can do those. Motor end plates, yeah, so it's a part of the skeletal muscle, the membrane of the skeletal muscle, the outer cell membrane, and the cell membrane of the neuron, right? The terminal bouton of the neuron, which is ending there. Right, so you have postsynaptic membrane and presynaptic membrane. Presynaptic is the nerve, postsynaptic is the muscle here, because it's a motor end plate or a neuromuscular junction, right? No, all primary cartilaginous joints don't get replaced by bone when growth ceases. Actually, those are the epiphyseal plates, right? However, the ribs, the costal cartilages in the front of the ribs, they don't get ossified. They basically stop about a few inches before the tip, and all the time they remain as cartilages, right? So they don't get ossified. It is the epiphyseal plates that get ossified. All of them get ossified, right? And fibrous joints, of course, not all the time, right? Practical book. The problem is there is no point in having a practical book, I suppose, because what practical book purpose is for you all to record the practical that you do, actually, things that you do in the lab. So there is no real reason for you to have a practical book at the moment, okay? Because what we do is we generally get you to draw all these things, name them, and, record the practical, basically, like you did in school. But now there is nothing, right? This is also just, I'm just doing this like a practical, but it is basically another lecture, right? So anyway, I'm doing this so that you will have some idea when you come for a practical. If we get the chance to do the practicals, we can quickly finish off, right? So that you have some knowledge and you can go through the practical quickly, okay? Skull with age, yeah. So sutures in the skull, they can ossify with age. As I said, ossification starts from the inner plate of the skull and continues towards the surface, right? So by gauging what are the sutures which have ossified, and again there may be differences between the sutures also, you can gauge the age of the skull, right? Fibrous joints. No, you don't call all the fibrous joints. You call sutural joints only in the skull, right? Only in the skull do we call them sutural joints. Okay, articular cartilage. Okay, I'll do that. Radial zone, chondrocyte orientation. Okay, I'll do the articular cartilage again. Cardiac muscle is branched. Yeah, I know, right? So the thing with slides is that. So when you make a slide, sometimes now PTH is made and you have to cut the slide and cut the muscle and do it. So sometimes you may not be able to see all the features in one particular slide, right? So sometimes branching may not be clearly visible, right? So it depends on the quality of the slide. Yes, so ground substance and matrix are the same thing, okay? So in the sense, matrix means whatever is outside the cells, right? So matrix actually, so matrix is made out of fibers plus the ground substance. Yes, that is true because, you can say that because ground substance is actually the proteins, right? So when you say cartilage matrix, you are including the fibers and the ground substance together, I suppose, because that is how it looks, right? So cells and matrix, okay? Cells and the matrix is made out of fibers and ground substance, right? But sometimes people just say the same thing for ground substance and matrix. But when you really think about it, it seems like the same thing also, but fibers and the proteinaceous material is definitely different, right? Okay. Chondroblast, chondrocytes. Yes. So cartilage and bone are all made by the cells, right? Chondroblast, chondrocytes, osteoblast, osteocytes, they lay down the bone. Except for mineralization in the bone, everything else is handled by the cells. They maintain the matrix, right? And they produce all the proteins and the fibers. Yes, okay. So I have already uploaded the morning lecture to Moodle. You can go through it, okay? If anybody has problems with connections, we always upload the video, so you can go through that, right? Okay, two primary cartilage between the manubrium and the first rib. So manubrium and first rib, two primary cartilaginous joints, right? Only in the first rib you get between the manubrium and the costal cartilage and the costal cartilage and the first rib, right? You're right. Bifurcating means dividing into two. Okay. Secondary cartilaginous joint, you have secondary cartilaginous joint, you have hyaline cartilage lining the joint surface. Then between the two joint surfaces, you have a disc of fibrocartilage, and inside that fibrocartilage, you can sometimes have a cavity, right? So if you're thinking about the pubic symphysis, oops, sorry, what happened? Just went up. No, right? Yeah. So if you think of the pubic symphysis, you have the two sort of pubic bones like this on either side, right? So this end will be lined by hyaline cartilage, and in the middle you will have a disc of fibrocartilage, and inside that disc, in the middle of that disc, you will have a small cavity, right? So that would be the cavity in a secondary cartilaginous joint, not like a synovial joint. Okay. Ground substance in connective tissue staining? No, not very clearly, because ground substance in anything doesn't stain clearly because it's just proteins, right? They don't stain well in HN. If you want to look at the ground substance, you can do special stains. That's why the special stain in the articular cartilage, the ground substance is specifically stained for a stain called safranino, right? So that intensity of that stain also tells you whether there is a lot of protein in that matrix or less proteins. Right? So it doesn't really stain in HNA that much. One lacune contain only one osteocyte. Yes. Okay. Costal cartilage and sternum, you get synovial joints, right? So costal cartilage and sternum, you get synovial joint. Costal cartilage and rib only, you get a primary cartilaginous joint. Classification of sutural joints? Not now, right? Past oh my God, I again forgot your papers. Yeah, there are books, right? There are many books, past paper books are there in the library. All the past papers are there in the library. You can ask one of your seniors to give you. They also must be having, I forgot again to bring that, okay, the past papers, because I don't really have them in my laptop. That's the problem, right? I will send you. I keep forgetting. I will put an alarm or something, right? So tomorrow. Okay. Right. Then anterior ramus from nerve plexus. What? Posterior rami also are there, right? So anterior rami are the ones that supply most of the skin in the anterior aspect. Posterior rami go to the back. Okay. They supply the skin and the muscles of the back. Okay. Mesenchymal cells and fibroblasts are quite similar. Mesenchymal cells are even more primitive, right? Fibroblasts are a little bit more mature than the mesenchymal cells, right? So you have these, I think in genetics, you will learn those. You pluripotent, totipotent, like that. Jayampati madam will teach you that. No, smooth muscle cell is not bifurcated. Lakshika madam will kill me if you tell that, right? There is no bifurcation in the, sorry, there is a slide. You can, at the tapering end, there can be small division, right? That's all. It is not branching, sorry. It's bifurcated at the end, right? So she will teach you, okay? Madam, all mucus secreting cells are goblets. I mean, you can, that is because of the shape, right? That you call it goblet cells. I mean, if it looks like a goblet, you can call them goblet cells, but it is okay to call them mucus secreting cells. Usually it's in the lung that we call them goblet cells, because that typical goblet appearance is seen in the lung, right? Sternocostal, yeah, so sternocostal joints are not primary cartilaginous, they are synovial, right? Syphisternal joint is a symphysial joint, secondary cartilaginous joint. Yeah, okay, so most of the time Snell says, apparently Snell says that it is, a joint is where the bones come together, though that's totally fine, right? So that is just going a little bit further and saying skeletal elements, because if you look at the classification, you see that there are some joints between bone and cartilage, right? And bone and fibrous tissue and bone, right? So only primary cartilaginous joint becomes a problem there, right? So most of it, 99%, is between bones, okay? Regarding brachial plexus, Snell says, I will check that and let you know, right? Why doesn't it say? Okay, I will check the Snell. Woven bone, no, there are no osteons, right? It's not lamellar bone, so just pieces of bone, like, okay? Myofibroblasts are not active, but I think they have the ability to contract also. They have muscular properties also, right? That's why you call them myofibroblasts. Not active in that sense, okay? They have ability to contract, okay? Right, so someone wanted that, this one, okay? So articular cartilage you wanted, was it? Yeah, so these are radial in the sense columns of cells, right? So you have a tangential layer. This is what you wanted, right? Okay, tangential layer on top, right? When the cells are like this. Okay, so this is the surface of the cartilage. So cells are like this. Okay, and then they become larger, slightly larger, and lie in groups. Okay, that is the transitional zone. Then they become even more larger and lie in lines like this. That is the radial zone, right? Then of course, again, you have groups where you have calcium deposition. Okay, is that what you wanted to ask about the articular cartilage slide?
Yes, madam.
Okay, right. All right. Yeah, can we come to the university? Yeah, we are really trying to get you down. Okay, we'll try because we have to have your seniors exam also and the hostels facilities again. The problem we have is because we give hostel facilities to most of the students. So because it's a residential university only, we have this big problem of bringing all of you down at the same time, right? So your seniors had a bit of a problem last time they came. COVID broke out, right? So because of that, several of your friends became, I mean, they were not badly affected. None of them were badly ill, just mild symptoms, but still, moment it comes into a hostel, because it's like an institution, it is a big problem, right? Anyway, we are working on it and we'll bring you down as soon as possible, right? So for now, I know it's difficult to be at home. So keep up your spirits, talk to each other. You can ask questions, you can talk to us if you want. Talk to your mentors, right? Message them and go through your things, right? So best way to deal with all this is to go through whatever you can initially, right? So the foundation ones are quite easy, but study them now itself and finish off and keep, so that you have more time to do limbs, right? Limbs is huge, right? A lot of details and a lot of new words. So I know we'll join condylosis. Yeah, so again, when you talk about different classifications, different shapes may be given different names, right? Metacarpophalangeal joints are, yeah, so condyloid means you have slight curvatures, not a flat, absolutely flat, but slight curvatures, right? So, yeah, it is a synovial joint, definitely a synovial joint, right? So condyloid is also another type that you can, according to the shape, you can call it that, and it is definitely a part of the synovial joint, right? So I think, not sure, some classifications will call it condyloid or some saddle condyloid and ellipsoid. I think those three may be similar in some classifications, right? So as I said, small differences can be there in the classifications. Don't worry too much about it. Okay. All right. Okay, so then we will stop. Now tomorrow you have this, yeah.